The human brain is wired to seek pleasure through dopamine, a neurotransmitter released in the ventral tegmental area and nucleus accumbens. These regions form the reward system, motivating behaviors like eating and social bonding. Pornography hijacks this system by delivering instant, intense stimulation, triggering excessive dopamine release. Over time, this artificial reward can rewire neural pathways, making natural pleasures less satisfying. Studies show porn activates these areas similarly to addictive substances, setting the stage for compulsive use. Natural rewards like food or exercise release moderate dopamine, encouraging balanced behavior. In contrast, porn provides hyperstimulation without effort, flooding the brain with 200% more dopamine than natural activities. This imbalance reduces sensitivity to everyday pleasures, a phenomenon called hedonic adaptation. Research indicates users often report diminished enjoyment in real-life intimacy after prolonged exposure to artificial stimuli. The brain begins to prefer the shortcut over harder-earned rewards. Neuroplasticity allows the brain to adapt structurally to frequent behaviors. MRI scans reveal that chronic porn users develop thicker gray matter in the dorsal striatum, habit center, but weaker prefrontal cortex connections, decision-making area. This physical change explains why quitting feels difficult. The addiction pathway becomes the brain's default. A 2014 German study found these changes mirror those in substance addicts, with neural alterations visible after just 5 weeks of regular use. Like drug tolerance, porn users experience desensitization. The brain downregulates dopamine receptors to cope with overstimulation. A Cambridge University study showed addicts require increasingly extreme content to feel the same excitement. This escalates into a vicious cycle. The ventral striatum becomes less responsive, while the amygdala, stress center, activates more strongly during attempts to quit. Functional scans reveal these users have blunted reactions to normal sexual stimuli compared to controls. Compulsion emerges when the habit loop overrides rational control. The prefrontal cortex, responsible for inhibition, shows reduced activity in addicts during craving episodes. A 2021 meta-analysis found 37% of regular users met clinical criteria for compulsive sexual behavior disorder, continuing despite negative consequences like job loss or broken relationships. Brain scans during cravings reveal patterns identical to cocaine addicts, with intense activity in the insula, craving region, and paralyzed executive control. The post-use crash involves dopamine depletion and serotonin disruption, fueling anxiety and depression. A Taiwan study tracking 23,000 men found porn users had 79% higher depression rates. Shame compounds this. The brain's default mode network activates intensely during self-criticism, creating a feedback loop. Neuroimaging shows addicts have heightened activity in the anterior cingulate cortex, pain processing, during withdrawal, explaining the emotional distress reported in recovery forums. Porn-induced erectile dysfunction, PIED, affects up to 30% of young men according to urology studies. The brain's sexual template gets rewired to respond only to exaggerated stimuli, making real partners seem unexciting. Couples therapy reports show partners of addicts often feel betrayed, reporting emotional distance. Neuroscientifically, mirror neuron systems, crucial for empathy, show reduced activation in heavy users during real intimacy, creating a disconnect that therapy must slowly rebuild. Today's high-speed internet delivers more sexual content in 10 minutes than our ancestors encountered in lifetimes. Data shows the average first exposure age has dropped to 11, while vanilla content represents under 5% of searches. Despite this, only 17% of parents discuss porn's risks with teens. Brain development research confirms early exposure increases addiction likelihood, as adolescent neuroplasticity makes habits harder to break. The normalization of extreme content creates generational desensitization. Effective recovery combines dopamine resets, 90-day abstinence with cognitive behavioral therapy to rewire associations. Exercise boosts prefrontal cortex regeneration, while mindfulness meditation thickens gray matter in decision-making areas. Support groups leverage oxytocin, the bonding hormone, to counteract isolation. A 2023 study showed combining these methods yielded 68% success rates at 6 months. Crucially, replacing the habit with healthy rewards helps rebuild the brain's natural sensitivity over 14 to 16 months. Neuroplasticity enables healing. After 18 months of abstinence, scans show near complete restoration of prefrontal control and dopamine receptor density. Recovered users report heightened appreciation for simple pleasures, from conversations to nature. A landmark study tracking 200 recovering addicts found their brain activity normalized to non-user levels. This mirrors stroke recovery timelines, proving the brain's remarkable adaptability. The journey is challenging but neurologically possible, offering hope for rewriting our deepest habits.